Welcome to chapter 10. So in this chapter we'll talk about lipids. So in this lecture we'll discuss lipids, fatty acids, waxes and triglycerols, chemical properties of triacylglycerols, and we'll discuss also phospholipids, steroids and cell membranes. So let's start with the skills that you require for this chapter. So you will need to understand how to write equations for hydrogenation, where you add hydrogen, hydration, adding water, and polymerization is where we talk about adding multiple bonds. And the next one is how to name alcohols and phenols, naming carboxylic acids, and finally hydrolyzing esters. So let's start with lipids. So what are lipids? Lipids are biomolecules. So they contain fatty acids or steroid nucleus. So the second thing is that they are soluble in organic solvents but not in water. So they are named for the Greek word lipos, which generally means fat. So they are an important feature in cell membranes, fat soluble vitamins and steroidal hormones. So let's discuss the types of lipids. So there are mainly 4 types of lipids. So we can characterize them based on their structure. This is an example of a fatty acid. So where you see a long chain carbon with a carboxylic acid at the end. So we call those fatty acids. So these are lipids like waxes, triacylglycerols, glycerophospholipids, and sphingolipids. So these are esters that can be hydrolyzed to give fatty acids and other molecules. So what are lipid containing glycerols? So there are two of them. So triacylglycerols and glycerophospholipids, they are structures that contain an alcohol, the glycerol. So you have a glycerol structure attached to fatty acids. And sometimes when you see glycerophospholipids, you have a phosphate group with an amino alcohol attached to that. So this is an example of lipids that contain glycerol. Sphingolipids contain sphingosine, which is an amino alcohol attached to fatty acid and a phosphate group and an amino alcohol. So these are ones that contain sphingosine. The other ones are steroids. So steroids which have a completely different structure, do not contain fatty acids and cannot be hydrolyzed. So we can characterize them by their steroid nucleus of containing the 4 fused carbon rings. You have 3 hexagonal carbon rings and 1 pentagonal carbon ring. So this is the base structure for any steroidal molecule. So an example of a steroid is testosterone, progesterone. These are examples of steroids, natural steroids in your own body. So what is the general structure of a lipid? So lipids, we can classify them simply into 2 types. One is fatty acid, the other is steroids. So fatty acids can be further classified into 4 types. You have waxes, triacylglycerols, glycerophospholipids, and sphingolipids. Steroids are a single class of compounds. Waxes, you have a long chain alcohol attached to a fatty acid. In triacylglycerols, you have a glycerol attached to fatty acids. In phospholipids, you have a phosphate and an amino alcohol attached to glycerol and fatty acids. In a sphingolipid, you have a sphingosine as the main branch and fatty acid on one of the stems and phosphate group on one of the stems. So stem is the sphingosine and the branches are the fatty acid and the phosphate group. So lipids are naturally occurring compounds. These are generally found in cells and tissues. So they are mostly insoluble. They are soluble in organic solvents, but they are not soluble in water. So let's try to answer this question. So lipids are characterized by the presence of fatty acids or a steroid nucleus. So which type is described by the following? So it contains a fused 4-membered ring system, contains long carbon chains and includes carboxylic groups. So which are characterized by the presence of, so which type follows this one here? So this is containing a 4-membered ring system. So this is a steroid. Contains long chain alcohols. So this is a fatty acid. And includes carboxyl groups. So this is a fatty acid. So this is how we can classify these structures. So pause the video right here and try to answer these questions. Next, let's discuss fatty acids. So the common one that we might think of is saturated fatty acids as chips with regular shapes that can stack closely in a can. So this is how you can think of as fatty acids. So there are some irregularly shaped chips that can be called, we call them the unsaturated fatty acids that do not fit closely together. So these are the two types of fatty acids that you will generally see. So fatty acids, they generally are the simplest type of lipids, and they contain long unbranched carbon chains with a carboxylic acid group at the end. They typically contain 12 to 18 carbon atoms long, and they are insoluble in water because of the long carbon chain. So if they do not contain a double bond, we call them saturated fatty acids. If they do contain a double bond, we call them unsaturated fatty acids. So if they do not have a double bond, saturated. If they have a double bond, unsaturated fatty acids. So how do you draw fatty acids? So in a line angle structural formula of a fatty acid, the end will always bend of the line that are carbon atoms. So we generally first draw the CH3 structure. We draw the CH2s as many as possible if it's a saturated fatty acid, and we write COOH. So these are the condensed structural formula, and we can also use the line angle structural formula, which is the line model. And the other one that can be represented easily is the ball and stick model. So ball and stick model is also another representation of a fatty acid. Now fatty acids can be saturated with only single bonds in the carbon chain. So if the number of carbons is 12 to the double bond, if the carbon atoms is 12, we call that lauric acid. Most commonly it's present in coconut. If it's 14 carbons long, we call it myristic acid. That is generally present in nutmeg. You have palmitic acid, which is the common substance in palm oil and palm. It contains about 16 carbons. In animal fats, you contain 18 carbon ring, so 18 carbon structure. We call that stearic acid. Most commonly it is found in animal fat. So notice that here, as the carbon size increases, the melting point also increases. This is due to the increasing dispersion forces. So increasing dispersion forces, which causes it to increase the melting point. So fatty acids can be monounsaturated with only one double bond in the carbon chain. For example, palmitoleic acid is generally the one that contains 16 carbons with one double bond, and most commonly it is present in butter, so it is palmitoleic acid. You have oleic acid, which is 18 carbons to one double bond, most commonly found in olives, pecan and grapeseed. So again, here also, as the number of carbons increases, you start noticing an increase in the melting point. So fatty acids can be polyunsaturated as well, with containing at least two double bonds in the carbon chain. So linoleic acid is the one that contains 18 carbons with two double bonds, generally found in soybean, safflower or sunflower. You have linoleic acid with three double bonds. So linolenic acid, which is three double bonds, it generally is present in corn. You have arachidonic acid, which is 20 carbons and 4 double bonds, which is generally found in meat, eggs and fishes. So here what happens, notice that the melting point actually decreases with the increase in the number of double bonds. So as you increase the double bonds, the melting point will reduce. So the more unsaturated it is, the lesser the stability of the structure. Now unsaturated fatty acids can be drawn as both cis and trans isomers. For example, if you take oleic acid, it is a monounsaturated fatty acid found in olives with one double bond at carbon 9. So if both side chains are written on one side, we call this a cis double bond. Now if both are drawn in a single line structure, straight structure, we call that a trans structure or a trans double bond. So this is the two structures that you notice here, how the differentiation work looks like. Now what are essential fatty acids? So humans are generally capable of synthesizing some fatty acids from carbohydrates or other fatty acids. You cannot synthesize sufficient amounts of polyunsaturated fatty acids such as linoleic acid or linolenic acid and arachidonic acid. So because these are polyunsaturated fatty acids, they must be obtained from the diet. That's why we call them essential fatty acids, because these are essential in the functioning of your body, but they cannot be directly synthesized by your own body, which you have to take it externally from your diet. So let's talk about the properties of saturated fatty acids. So saturated fatty acids contain only single carbon-carbon bonds and fit really close together in a regular pattern with really strong dispersion forces between the carbon chains. And they require a significant amount of energy and high temperatures to separate them and melt them. So in terms of in unsaturated fatty acids, the cis double bonds cause the carbon chain to bend or kink, giving the molecule an irregular shape, thus allowing for fewer interaction with the molecules. So trans structures are much more similar to the original structures. So this reduced interactions here in fatty acids with cis bonds reduce the melting point of the molecule. This is one of the reasons unsaturated fatty acids are solid in terms of they are liquids at general temperatures and they cannot sustain longer. So pause the video right here and try to answer the questions below. Next, let's talk about prostaglandins. So prostaglandins are hormone-like substances that are produced in really small amounts in most cells. So the other name is called eicosanoids. So they are generally formed from arachidonic acid, a polysaturated fatty acid, with 20 carbon atoms. They generally differ by substituents attached to the 5-carbon ring, and they have many functions such as lowering or raising blood pressure, stimulating contractions, and relaxation of the smooth muscles of the uterus. So prostaglandins, they generally differ by their substituents that are attached to the 5-carbon ring. So we have prostaglandin E, which has a ketone group on carbon 9, and prostaglandin F, which has a hydroxyl group on carbon 9. And the number of double bonds is shown as a subscript as 1 or 2. For example, this structure is PGE1 because it has one double bond, and it is E because there is a ketone group on the 9th carbon. Here you have a hydroxyl group on the 9th carbon and a double bond, single double bond, so which becomes PGF1. Now, when tissues are injured, this arachidonic acid is converted into prostaglandins that produce inflammation and pain in the area. So arachidonic acid creates PGF2 or PGE1 and PGF1 depending on the reception that is received. So non-steroidal anti-inflammatory drugs or NSAIDs, they generally block the production of prostaglandins and they decrease the pain and inflammation. So most common out of them is ibuprofen that you generally take when you feel pain. So that pain production is due to the arachidonic acid because analgesics, as these are called, stop the production of conversion of arachidonic acid into PGF2, which generally causes pain, fever, or inflammation. Now, NSAIDs are generally include naproxen, which is in common name is Aleve or naproxen. You have ketoprofen, which is Actron, and you have nabometone, which is Relafen. You have aspirin, ibuprofen, and naproxen. These are the common compounds that you generally see. So next, let's talk about omega-6 fatty acids. So unsaturated fats are those that are in vegetable oils and fish. They are recognized as more beneficial than saturated acids. So vegetables generally contain omega-6 acids, meaning that the first double bond that you see is at the 6th carbon. So from the methyl end of the carbon chain. So examples of omega-6 acids are linoleic acid and arachidonic acid. So these are the two acids that are generally called as the unsaturated acids. And out of them, the omega-6 fatty acid is the linoleic acid. Now, why are omega-3 fatty acids much more important is that fish in general have high levels of omega-3 acids. So meaning that the first double bond occurs at carbon 3. from the methyl end of the chain. So examples of omega-3 is linoleic acid, pentanoic acid, docosahexaenoic acid. These are examples of omega-3 fatty acid, omega-3 acids. Now, in two diseases mainly, atherosclerosis and heart disease, the cholesterol forms plaques that adhere to the walls of the blood vessels. Now, these blood pressure in general rises as the blood fluids through small openings in the blood vessels. Now, when more plaque forms, there is also a possibility of blood clots blocking the blood vessels and causing a heart attack. So, omega-3 fatty acids can lower the tendency of blood platelets to stick together and reduce the possibility of blood clots. So pause the video right here and label each of the following acids as saturated, monounsaturated or polyunsaturated. Next, let's go to the topic of waxes and triglycerols. So waxes and triglycerols are natural waxes, are generally found on the surfaces of fruits and on the leaves and the stems of the plants. So where they help prevent the loss of water and damage from pests. So waxes on the skin, fur and feathers of animals provide what we call as a waterproof coating. So let's start with waxes. So waxes are generally found in both animals and plants. So they are found on the surface of fruits and on the leaves or the stems of the plants, so which help them from losing water and damaging from the pests. So they are found on skin, fur, feathers of animals and provide as a waterproof coating. Now some examples of this is beeswax. So beeswax contains a long chain structure. So these are esters. Remember that these are long chain esters. So they have, generally the source of beeswax is from the honeycomb, and it's used for candles, shoe polish and wax paper. You have carnauba wax, which is also the same kind of ester. It is generally, the source of it is from the Brazilian palm tree. It generally is used for waxes for furniture, cars, floors or shoes. Next you have jojoba wax, so which is also an ester which contains generally from the jojoba bush. It's used to make candles, soaps and cosmetics. So in the blood, fatty acids are generally stored as TAGs or triglycerol or triacylglycerols, or in simple words, triglycerides. So which are generally esters of glycerol, a trihydroxy alcohol and a fatty acid. So these are formed when you have 3 hydroxyl groups of glycerol that react with the 3 carboxyl groups of the fatty acids. So what happens here, glycerol has the following structure. So this is the example of glycerol. Now if one of the protons goes out and is replaced with a fatty acid in place of that proton, attach it to a carbonyl group, so that becomes an ester. So the ester in turn creates the triacylglycerols. So these we can change by glycerol to glyceryl, and we can name it as fatty acids as the carboxylates. So for example here, this is an example here, the oxygen and the CH carbon part of it stays on one side and the fatty acid part of it stays on the other side. So the simple representation is this representation to write a glycerol molecule. So the general formula for triacylglycerol contains 3 hydroxyl groups of glycerol forming ester bonds with carboxyl groups of 3 fatty acids. Now glycerol and the 3 molecules of stearic acid form triacylglycerol. Remember that these acids are from stearic acid. Now glycerol is named after glyceryl and the fatty acids are named as carboxylates. Now to name the fatty acid formed with 3 stearic acids, the name becomes glyceryl tristearate, and the common name for this compound is tristearin, and this is where you see the formation of the 2 parts in the glycerol and 3 stearic acid molecules form glycerol tristearate or tristearin. Now we can also find mixed triglycerols. So these are ones that contain different fatty acids rather than having a single fatty acid. So for example, you can make it from stearic acid, oleic acid and palmitic acid. So this is an example of a mixed triglycerol where you have the glycerol structure stays the same, but the acids that are attached to them will differ. So these are examples of triacylglycerols, mixed triacylglycerols. Now triacylglycerols are the ones that store energy This is the major form of energy storage in animals. So animals that generally hibernate eat large quantity of plants, seeds and nuts that are high in calories. So as the external temperature drops, the animal goes into hibernation and the body temperature drops to nearly freezing, and it reduces the cellular activity, respiration and heart rate. Now, triacylglycerols, in terms of the energy storage, so during hibernation, their stored fat is in the form of triacylglycerols, which is their only source of energy. Now, pause the video right here and try to name the fatty acids that are connected to the glycerol molecule here. Now, so let's talk about the guide to drawing triacylglycerols. Step 1 is to draw the condensed structure of the formulas for glycerol and the fatty acids. And finally, form the ester bonds between the hydroxyl group of glycerol and the carboxyl groups of the fatty acids. So remove OH and H. So remember that this OH and H go out as H2O. This is dehydration reaction. And each of these will yield 3 hydrogen, 3 water molecules. And the combined compound is what creates the acid. So when the combination, you get the triacylglycerol. So here we have glyceryl structure and you have a palmitic acid structure, so it becomes tripalmitate. So now draw the condensed structural formula for triacylglycerol containing 3 molecules of myristic acid. So you already have the myristic acid from the last slide, so use that structure and try to draw the condensed structure. Next, what are the melting points of fats and oils? So a fat in general is usually solid at room temperature. It usually comes from animal sources such as meat, whole milk, butter or cheese. And oil is generally usually liquid at room temperature and is usually obtained from a plant source such as corn or olive. Now, oils with unsaturated fatty acids, for example, oils that form from olive, peanut are generally monounsaturated because they contain large amounts of oleic acid. Now, corn, cottonseed, safflower seed, sunflower seed are polyunsaturated because they contain large amounts of fatty acids with 2 or more double bonds. Now, palm and coconut are solids at room temperature because they consist mostly of saturated fatty acids. Now, saturated and unsaturated melting points depend on the saturation of the fatty acid. Now, saturated fatty acids generally will have higher melting points than unsaturated fatty acids because they can pack more tightly together, and they are generally found in animal fats, coconut oil and palm oil. So this is a diagram that shows the percentage of saturated and unsaturated fats in different oils. You notice that coconut oil contains a lot of saturated fat, and you can see that olive oil contains a lot of monounsaturated fat, and you can see that safflower oil contains a lot of polysaturated fat. So the ones that contain a large amounts of polysaturated fat become polyunsaturated fat, polyunsaturated acids. So these are different vegetable oils. Next, let's talk about the chemical properties of triacylglycerols. You have many soft margarines, stick margarines, and solid shortenings are generally produced by partial hydrogenation of vegetable oils. So we take vegetable oils and we do partial hydrogenation that creates the margarine, which is generally used as a replacement for butter sometimes, and that is something that you can do as well. So in hydrogenation reactions, what will happen is that the double bonds in unsaturated fatty acids react with the hydrogen gas to produce carbon-carbon single bonds, and the hydrogen gas is bubbled up through heated oil, typically in the presence of a nickel catalyst. So this is the process of hydrogenation. So next, let's talk about hydrogenation reactions. So what do we do in commercial hydrogenation? So we add hydrogen and it stopped before all the double bonds in a liquid oil become completely saturated, and partial hydrogenation of a liquid vegetable oil changes it to a soft semi-solid fat. So the more saturated the product, the higher the melting point. So pause the video right here and try to judge what product will you get when you do complete hydrogenation of glyceryl trioleate. Now, in terms of the link to the health, so hydrogenation and interesterification. So unsaturated fatty acids can be both cis and trans. So which means that during hydrogenation, you can have double bonds that are converted to single bonds, and a small number of cis double bonds are converted to more stable trans double bonds, causing a change in the overall structure of the fatty acid. Now, in the body, these are called trans fatty acids. They behave like saturated fatty acids and are estimated to be 2 to 4% of our calories. So they raise the LDL cholesterol and lower the HDL cholesterol, which is this LDL cholesterol is what creates plaque in the blood vessels. So the higher the amount of LDL cholesterol, the higher the chances of a risk of a stroke. So this is the reason why you should avoid trans fats. So this process of hydrogenation creates the trans fatty acids, where the cis fatty acid under hydrogenation can create an undiarisable side product, which is the trans fatty acid, and the desired saturated product. Now, what are some of the foods that can contain these is milk, eggs, and beef. So food that contains trans fatty acids from hydrogenation process are deep fried foods, bread, baked goods and cookies, crackers and chips, stick and soft margarines, and vegetable shortening. So these are some of the food that contain fatty acids due to the process of trans hydrogenation. So pause the video right here and try to answer this question. So pause the video again here and try to judge what are the two products that you get from this reaction here. Next, let's talk interesterification. So interesterification is a newer process. It is generally used to change unsaturated vegetable oils into products that have properties of solid and semi-solid saturated fats. So they are used to hydrolyze ester bonds in triacylglycerols and of fatty acids using lipase enzymes, so that the glycerol and the fatty acids are generally formed, and the saturated fatty acids then recombine with glycerol. So in interesterification, we use the enzymatic reaction to make sure that the process, you have an unsaturated oleic acid in a vegetable oil replaced by a saturated stearic acid. Remember that we are not altering the double bonds, we are actually changing the entire structure of the carbon skeleton itself. Next, let's talk about hydrolysis. So triacylglycerols can be hydrolyzed, which is meaning that you can split it with water in presence of really strong acids such as HCl and H2SO4. The most common are the other compounds, which are the digestive enzymes, which we call lipases. Now, the products of ester hydrolysis are generally glycerol and 3 fatty acids. Now, the polar glycerol is soluble in water, but the fatty acids and their long chain hydrocarbons are not. Now, what happens when you add water to ester bonds is that it splits it and creates glycerol and the 3-palmitic acid molecules. So here we are using tripalmitin that creates glycerol, and so this reaction is hydrolysis reaction. Now, so with that we end lecture 1 of lipids. So in the next lecture we'll continue the topic on saponification.